Health and co-chair of FDA Omics Days. We feel that this meeting is at the core of the Omics Working Group because it is based on its main objectives. Established in late 2021, the Omics Working Group evolved from the original Genomics Working Group to encompass the broader spectrum of omics technologies. The Omics Working Group consists of 4 specialized subsections: the Genomics, Transcriptomics, and Metagenomics subgroup, the Proteomics and Metabolomics subgroup, the Data Integration and Data Management subgroup, and the Omics One Health subgroup. This meeting serves as a platform to discuss the latest advancements in the field and to further the mission of the FDA Omics Working Group to increase awareness of omics activities at the FDA and to provide expertise in omics data analysis techniques. By gathering scientists and reviewers from across various FDA centers, we aim to share experiences, resources, and innovative ideas. This symposium was organized in part from the Omics Working Group members' recognition of the growing use of omics and the need for a dedicated forum to explore this cutting-edge field. Today, we will learn about the current applications of omics in academic and regulatory environments and examine the challenges. We are honored to kick off our symposium with opening remarks from our FDA Commissioner, Dr. Califf. Following that, we have our keynote address by Dr. Archana Chatterjee from Rosalind Franklin University. The day will continue with a series of breakout sessions, starting with the challenging landscape of precision medicine and pharmacogenomics, followed by omics applications on safety and efficacy of product development, and concluding with leveraging omics for surveillance, emerging threats, and One Health. We would like to take a moment to acknowledge the invaluable contributions of our FDA Omics Days co-chairs, Drs. Paula Hyland and Richard Beeger, our steering committee and session chairs, whose efforts truly made this event possible. As a reminder, please ensure that your cell phones are on silent and save your questions for the end of each session. You may find breakfast and lunch items at the kiosk. For virtual attendees, please use the Q&A box in Zoom to submit your questions at any time during the session. After each keynote or session, the moderator will prioritize in-person questions followed by online questions from the Q&A box. If speakers are available, we welcome them to address the remaining questions from the Q&A box after the session ends. Today, we are honored to begin our symposium with opening remarks from our Commissioner, Dr. Robert Califf. Dr. Califf was confirmed as the 25th Commissioner of Food and Drugs. He also served in 2016 as the 22nd Commissioner and immediately prior to that as the FDA's Deputy Commissioner for Medical Products and Tobacco. He has spent a good portion of his career affiliated with Duke University, where he served as a professor of medicine and vice chancellor for clinical and translational research, director of Duke Translational Medicine Institute, and was the founding director of Duke Clinical Research Institute. He has had a long and distinguished career as a physician, researcher, and leader in the fields of science and medicine. He is a nationally recognized expert in cardiovascular medicine, health outcomes research, healthcare quality, and clinical research, and a leader in the growing field of translational research, which is key to ensuring that advances in science translate into medical care. And now, I will hand it over to Dr. Califf for his opening remarks.
Thanks for that kind introduction, and I sure agree with Long about the career. I'm not so sure about distinguished. That's a matter of opinion. I was reminded of that Duke is celebrating its centennial this year, 100 years at the university. I arrived as a freshman in 1969, so I've been affiliated with the university for over half of its entire longevity. I'm not sure what that means, but I'm also sporting my USA tie today. I was very inspired last night. I went to the Sammy Awards event, which, as many of you know, is recognizing through a very interesting selection and voting process the meritorious service of civil servants. Out of the 3 million total people working for the U.S. government, they end up with a small group. Janet Woodcock was honored there, who many of you know as a finalist for the Distinguished Career Award. But just recognizing what government people contribute to this country all at one time in an event like that was remarkable. So I am pleased to kick off the Omics conference here, and I have some prepared remarks. You know, the term omics, so seemingly simple and straightforward, is profoundly complex, as you all know. It involves and embraces a wealth of disciplines, subjects, and scientific developments that belie the seeming surface simplicity. Wikipedia provides a common-sense definition. Omics aims at the collective characterization and quantification of pools of biological molecules that translate into the structure, function, and dynamics of an organism or organisms. This construction of working models of how biology works at multiple levels is about as complicated as it gets. In this way, omics almost seems to mirror the FDA itself, which also encompasses a broad range of disciplines, responsibilities, and authorities that we're tasked with in our scientific and regulatory mission to protect and advance public health. We also engage in an extraordinary and diverse array of talented scientists, regulators, and others who apply a wide range of expertise to address these challenges. Furthermore, like the FDA, many of the areas of focus within omics are topics at the forefront of scientific advancement today, and I think you see that in the program. They include subjects such as next-generation sequencing and mass spectrometry, which find their omics in fields like genomics, transcriptomics, metagenomics, proteomics, metabolomics. It goes on and on. These, along with many other disciplines within the categorization of omics, are used by scientists across the FDA for addressing the many distinct, challenging, and highly technical areas of oversight for which we're responsible, ranging from the discovery and development of new medicines to address treatment of diseases to the surveillance of pathogens and the ability to track and respond to outbreaks of foodborne illness, and so much more. In all of these areas, as well as combinations of them, we're learning more and more about the interplay of different biological components and gaining greater understanding of the ways for us to respond to threats against the health of humans and animals. But the world of omics also involves more than individual scientific discoveries or advances or even the exciting explorations of new areas of science. It also includes and relates to how we measure and interpret these subjects through the broader use of diverse data sets, which has contributed to improvements in data analysis, data interpretation, and data visualization. As our computational capacity expands and the tools and instruments we use to measure and analyze complex multidimensional biology, behavior, environment, and social interactions grows, along with digitization and our increased ability to share this information, the opportunities we have to explore and find answers to questions about the human condition and the world in which we live grows as well. And increasingly, we have new technologies such as artificial intelligence and machine learning tools to support us in this aspect of our work in omics as well. So the scientific and computational underpinning of our work is accelerating at an astounding pace. It should come as no surprise that the most recent report from the White House, earlier updating critical and emerging technologies, recognized omics as a key aspect of this body of science. At the FDA, of course, we've known this for some time, and your work in this group is a critical component of regulatory science. There's so many different ways in which omics-related work by FDA employees helps protect the public's health, from pathogen and disease surveillance to the development of new vaccines, therapies, and diagnostic tools. So much of the work we do relies on our ability to be at the cutting edge of science so that we stay ahead and support industry in the development of new products and provide the public with the public health protections they need. I want to commend the FDA Omics Working Group for its continuing efforts in this area to bring together researchers and staff from across FDA to facilitate collaboration and sharing of information and ideas. The group has provided education and training opportunities for FDA staff through the hosting of symposia like this and seminar series featuring external subject matter experts, and regularly contributes its knowledge and expertise to FDA-wide events such as the annual FDA Scientific Computing Days. These opportunities have helped increase the capabilities of FDA staff by introducing them to rapidly developing changes in omics tools and by contributing to modernizing FDA and filling existing knowledge gaps by ensuring FDA is ready and prepared for the introduction and use of new technologies. The working group also supports FDA reviewers by providing consultations, recommendations, suggestions, and scientific advice on sponsor-submitted omics data and applications for new products and therapeutics. Today's conference is yet another example of how the Omics Working Group demonstrates leadership in this area by bringing scientists and researchers together to share data and information that's needed for the FDA to fulfill its mission to protect and advance public health. I think the title of this year's meeting, Precision in Practice, Regulatory Science, Best Practices, and Future Directions in Omics, truly captures the promise that omics brings to our workplace. The three sessions will provide some far-reaching opportunities for discussion of some of the ways that omics can change and is changing medical research and the practice of medicine, and help provide a critical perspective and roadmap for the work we do. Session one, as just described, will explore some of the promises that precision medicine offers for improved health by accounting for individual variability in genes, environment, and lifestyle. As we know, researchers today are increasingly and routinely using healthcare data to identify the genomic underpinnings of cancer and many other diseases, both common and rare. They're introducing transformative, molecularly targeted therapies using an individual's genetic profile and pharmacogenomics to make the best therapeutic choice by facilitating predictions about whether that person will benefit from a particular medicine or suffer serious side effects. And they're leveraging massive computational capabilities with new machine learning methods. All of these advances are beginning to generate tangible results from the potential contribution of different omics technologies or data to the identification of predictive and prognostic biomarkers and the development of targeted therapeutics. Keynote speaker for this series, Professor Chatterjee, will address the potential role of precision medicine in infectious diseases and vaccine development that may lead to the development of safer and more effective vaccines. She'll highlight how the availability of large omic and clinical data and appropriate application of artificial intelligence may allow precision in vaccines and public health. This session will also offer experiences from a number of academics and experts in their fields about the practical applications of multi-omics, artificial intelligence, and precision medicine in rare disease and oncology research for the identification of modifiers and predictors of disease, onset, severity, and survival, which can lead to a better understanding of the pathogenesis of disease and the development of suitable therapeutic strategies. Session two focuses on omics applications on safety and efficacy of product development. The bioinformatics analysis of omics data increasingly is being used to assess product quality, safety, and efficacy to facilitate the development, characterization, and manufacture of drugs, devices, and biologics. The most recent iteration of PDUFA 7, which was reauthorized last year, recognized a substantial increase in volume and diversity of bioinformatics information and data in regulatory submissions, such as next-generation sequencing, and consequently, it included bioinformatics-specific goals. This session will include presentations by academia and FDA scientists exploring the use of mass spectrometry-based omics to identify health hazards associated with the use of tobacco products. It will also include the use of omics in vaccine efficacy studies, including identification of endogenous viruses and multi-omics-based computational models of immunity, and the use of multi-omics in understanding safety and efficacy of xenotransplantation, an exciting and emerging field with multiple live human transplantations having now been performed. Finally, Session 3 takes a look at leveraging omics for surveillance, emerging threats, and One Health. As you're probably aware, the FDA's One Health initiative works to build and support effective, collaborative, interdisciplinary strategies to protect human and animal health based upon the essential connections that exist among the health of people, animals, and the environment. While this has wide-ranging applications, it has particular significance for the two areas that this session will focus on: antimicrobial resistance and food safety. The FDA partners with other federal agencies in a collaborative effort to detect, prevent, and limit the impact of AMR through the development of enhanced diagnostic and surveillance tools and new drugs and biologics to prevent or treat infections. And when it comes to food safety, the FDA oversees about 78% of the U.S. food supply, which involves continuous surveillance of ongoing and emerging threats, development of methods for countering these threats, and deploying these methods to domestic and global partners. The presentations of this session will highlight the use of NGS-based tools for the environmental surveillance of infectious diseases and AMR, biothreat surveillance, and food safety. Speakers include subject matter experts in omics technologies who collaborate with experts outside of their respective fields, such as agricultural scientists, regulatory scientists, epidemiologists, microbiologists, and policymakers. This aligns with FDA's One Health initiative focus on fostering collaborations and a holistic approach to achieve optimal health outcomes for people, animals, plants, and their shared environment. As I've been dealing with the H5N1 outbreak in dairy cattle, I'm convinced that we need to double down on One Health and move beyond the rhetoric into more effective action. There's a lot of work to do on this topic. I'm particularly looking forward to some events at Cornell and out in Colorado that we're planning to focus and bring attention to One Health to the broader political and lay community, because we have a real topic to focus on in H5N1 that a lot of people are concerned about. I hope this brief summary makes clear just how jam-packed and stimulating a day that's planned today. It's an opportunity for you to learn, to share, and to collaborate in ways that will lead to further development of innovative solutions to medical and scientific challenges. It offers a chance for you to work across disciplines and develop ways to provide employees with new tools, approaches, and passion for their work. Before I send you on your way so that you can immerse yourselves in these exciting topics, I want to briefly reiterate and expand upon a point of caution I mentioned last year based on my own experience in this field. There is no doubt the potential offered by the science of omics is enormous, and the science itself is awesome and exciting, providing both professional opportunities and a chance for extraordinary medical and scientific advances. And yet we cannot simply treat this work as if we're children in a candy shop. We must remain diligent in the oversight of the scientific enterprise and work to ensure that the conclusions that we reach are well supported and the potential for error, whether accidental or even in some cases intentional, is minimized. As I've had the privilege of working in omics studies in my own research and overseeing such research in academia and in a big tech company, I feel it's important to note that one quality of this arena is that each time the technology improves, our understanding of how the array of molecules and systems interacts changes. This quality of the science causes me to stress the importance of communicating to the public about this work as it evolves. The complexity and rapid evolution of the science gives us a difficult task to explain what you're doing and what you're working on to achieve. The public is, after all, the ultimate beneficiary of this work. Scientists today increasingly face a growing dissemination of misinformation about the work you do, about science in general, and even more directly about the validity and value of science done in agencies working to protect the public health, such as the FDA. Indeed, this misinformation is being used to undermine public confidence in science generally and biomedical science in particular. The result threatens to subvert the credibility of our agency's work, as well as promote the continued decline in health status of this country. As I'm sure you're all well aware, we're now pretty much at the bottom of the heap in terms of life expectancy for high-income countries. Who would have thought that despite our tremendous knowledge about biomedical science and our ongoing success in translating that knowledge into effective medical products, we're behind most other, and in fact almost all high-income countries in our health status, and the gap is growing. So as you engage in discussions and strategies about groundbreaking science, I encourage you to think about three related points. First, we need to do a better job of communicating with the public in plain language whenever possible, to explain what we're doing, why we're doing it, and what it means, particularly with respect to informing decisions about health. And here I'll just note there's a real tendency, because of the excitement, to overstate the claims related to complex genomic analyses as they relate to human health. Second, we need to produce the most reliable scientific work possible by using the most scrupulous and sophisticated scientific, investigative, and regulatory methods and tools to achieve this result. Those who would seek to undermine the trust of the American public in science and public health will capitalize on any shortcomings that are revealed. It's worth noting that our responsibility to the public is difficult and seemingly conflicted. We must communicate uncertainty even as we work to gain and maintain public trust. Clear and accurate communication with the public and product developers based on sound scientific principles and empirical evidence will support increased trust and reliance on the integrity of our work. And here I have something new that I discovered yesterday. Francis Collins has his next book, which is about to come out. There's a very good interview by my former colleague, Eric Topol, with Francis Collins, and I think it's going to be a good book to read about this very topic of how to deal with the public. And he talks about his own shortcomings during the recent pandemic in terms of understanding how to transmit uncertainty in the face of evolving science. Finally, we employ collaborative methods to combat misinformation in all of our scientific work, working together through coalitions with partners across HHS, as well as with trusted messengers in external scientific and public health organizations, educators, and clinician groups, is an essential aspect of communication to the benefit of the public. The challenge is broader than FDA, and so is the solution. This brings me back to this meeting and the important work you all will be doing here. Each of the three principles I outlined, communication, scientific accuracy, and collaboration, are an important component of your efforts, and they also are at the core of the work being done in omics at the FDA. As you share information and ideas during this meeting, as you bring together researchers and staff from across the agency, and as you build coalitions, increase communication, and strengthen our work across our centers, I know that you'll put these approaches into play to produce a product that makes the FDA stronger and more effective. So thanks for your commitment to public health. Thanks for being here, and I hope you have a productive and inspiring meeting. I'm going to have to run, but appreciate the chance to interact with you.
Thank you, Commissioner Califf, for those opening remarks. Well, good morning, everyone. My name is Paula Hyland, and I'm a staff fellow or senior staff fellow and genomics lead in the Division of Applied Regulatory Science in the FDA's Center for Drug Evaluation and Research. I'm a molecular biologist and a molecular epidemiologist, and at the FDA, my job functions include genomic support for reviews in rare diseases and research focusing on biomarker discovery, including omics technologies, for drug response and toxicity. So this morning's keynote address will be given by Dr. Archana Chatterjee, who is the Dean of the Chicago Medical School, Senior Vice President for Medical Affairs at the Rosalind Franklin University of Medicine and Science. And if you would please go ahead, Dr. Chatterjee, and begin sharing your slides while I continue with the introduction. Dr. Chatterjee joined Rosalind Franklin University in April 2020 and is the first woman and woman of color to serve as Dean of the Chicago Medical School and Senior Vice President for Medical Affairs. Prior to this, she was also the first woman and woman of color to serve as Professor and Chair of the Department of Pediatrics and later Senior Associate Dean for Faculty Development at the University of South Dakota, Sanford School of Medicine. At Creighton University School of Medicine, she advanced through the academic ranks to tenured professor. She served as division chief and the hospital epidemiologist at the Children's Hospital and Medical Center in Omaha, Nebraska. Dr. Chatterjee has also been elected and served on several national advisory boards and committees, including the FDA's Vaccines and Related Biological Products Advisory Committee, the Association of American Medical Colleges Group on Women in Medicine and Science, the Pediatric Infectious Diseases Society Board of Directors, and the American Board of Pediatric Subboard of Pediatric Infectious Diseases. Having trained as a pediatric infectious disease specialist, Dr. Chatterjee has practiced in her field for over 20 years, conducted over 120 clinical trials, published over 100 peer-reviewed articles and many invited review articles, and 31 book chapters and 1 book. Dr. Chatterjee, I'll hand it over to you now, and thank you for joining us today.
Thank you very much. Good morning. I hope you all can hear me. I would like to begin by thanking the organizers for inviting me to give this keynote address. And I would also like to ask the audience to take a moment to reflect on the colleagues, family, and friends who support you in your life and endeavors. So please take a moment. Thank you. With regard to disclosures from my presentation, I have none. I included this photo in my presentation of Dr. Rosalind Franklin's mural that stands at the entrance to our university, because I think, especially for this Omics Day discussion, recognizing her work in the discovery of the structure of DNA is critical to really where we are at today, particularly in the field of genomics. So I would just like to acknowledge that at this point in time. So quickly, the outline of my talk today. I know that this is a very savvy audience, but I also know that there may be some members in the audience that perhaps are not as familiar with precision medicine. So I'm going to start with the definition of precision medicine. I'll then discuss the role of precision medicine in infectious diseases and spend most of this talk describing the role of precision medicine in vaccinology, which is really my area of expertise. I will then define precision public health and talk at the end about how we can rebuild trust in government, healthcare, and science through precision medicine. You will see throughout my talk that there are a number of cartoons. I love cartoons, so I include those. One thing to keep in mind when we are talking about omics and precision medicine is that the bulk of the work has been done in the therapeutic world, particularly in the field of oncology. So a search of papers related to this field shows that nearly 40% of them are from that field, only about 4% from the infectious diseases world, and less than 1% that are focused on infectious diseases and vaccinology. So what is precision medicine? It's also been described as personalized medicine. And I decided to use the FDA definition, which is an innovative approach to tailoring disease prevention and treatment that takes into account differences in people's genes, environments, and lifestyles. And it's the prevention aspect that I think has not received as much attention as perhaps treatment has. In lay terms, precision medicine can be thought of as giving the right treatment at the right time, every time, to the right person. As I mentioned, most of the scientific literature is in oncology and based on treatment and genomics. But when we look at the role of precision medicine in infectious diseases, it behooves us to remember that infections lead to a large proportion of the morbidity and mortality seen around the world. The WHO lists infections as one of the top 10 global health concerns. And this is because infections not only exist among us continuously, but emerge and spread rapidly. And as we know from the recent experience with COVID, that this can impact the whole world. As I mentioned, not only can they cause morbidity and mortality, but they can actually cause significant disruption in our lives, economic damage, et cetera. It's also important to remember that infections contribute to cancers. It's estimated that about 13% of all cancers are actually caused by some type of infectious agent. The one best known for it is human papillomavirus, or HPV, where a third of all infection-related cancers are actually caused by HPV, and it is the fourth most common cause of cancers in women. We also have known for a while now that hepatitis B virus is associated with hepatocellular carcinomas. So let's turn to COVID for a minute and talk a little bit more in detail about some biomarkers and risk factors for COVID-19 severity, because we know that this is an infection that actually spreads across many different levels of disease. So there are people like myself who probably have been infected, but despite being tested multiple times, have never tested positive. So had essentially asymptomatic infection. And then there are others, like 3 of my uncles, who became severely ill and died from COVID. What this has to do with is a number of different factors that you see listed on this slide, including genetic variations, ACE2 polymorphisms, and I'll talk a little bit more about the role of ACE2 here in a minute, non-O blood types, advancing age, which probably was the factor for my uncles, other comorbidities, obesity, diabetes, cardiovascular diseases, immune compromise, et cetera. We know that racial ethnic background has had a role to play in that, at least in the United States, people from underrepresented groups have had a disproportionate impact from this virus, from this disease. And while some of the above mentioned factors may play into that, some of it also has to do with their socioeconomic status, their access to care, et cetera. So let's hark back a little bit to how SARS-CoV-2 actually causes disease. And it has to do with the renin-angiotensin system. So a little bit of a reminder on this. You all know probably that angiotensin 1 gets converted by the angiotensin converting enzyme to angiotensin 2, which through a couple of cellular receptors in the lung can cause vasoconstriction, inflammation, fibrosis, lung damage, and edema. The angiotensin 2 converting enzyme 2 actually then takes angiotensin 2 and converts it into angiotensin 1 through 7, which through another cellular receptor actually causes vasodilation, protection, prevents lung damage, and inhibits edema. SARS-CoV-2, the virus that causes COVID, attaches to this receptor and actually downregulates it, which then results in overactivation of the renin-angiotensin system and causes the damage in the lungs that we see. Now, it's interesting to note that the risk factors and the impact of those and outcomes vary tremendously, as you see listed on this slide. So I mentioned non-O blood types. There's a slight increase in infection prevention about people from those blood types. But among those blood types, there are differences in intubations, risk of death, and those are listed in this first bullet. We know that the elderly, people over 65, made up 92% of all of the deaths from the virus. So that's the impact of age. Obesity, we know, impairs immune function, decreases lung capacity and reserve. And I talked just now about the risk in the racial and ethnic minority population of Hispanics and Blacks. We know that people with diabetes are at increased risk of morbidity and mortality due to COVID. And interestingly enough, COVID also increases the risk of developing diabetes. Patients with cardiovascular disease had a 3 times higher risk of critical illness and twice the mortality of non-CVD patients with COVID. We also know that COVID increases the risk of myocarditis, pericarditis, and AFib. Immunosuppression was noted with a 3-fold increase in the risk of severe COVID disease, and people with HIV have a 38% increased risk of developing severe or fatal COVID. I did mention socioeconomic status just now, but it's interesting to note that studies have shown that lower education level is actually the most strongly associated with cases and fatalities. Turning to HPV, it's estimated that 12% of women worldwide are infected, and the oncogenicity, of course, depends on the viral strain as well as patient epigenetics. The most common oncogenic types are listed here, with 16 and 18 being highlighted because they cause about 70% of all the cancers caused by HPV. What HPV does is induce modifications in the host cellular chromatin, including DNA and histone methylation, which results in the aberrant expression of genes. And twin and family studies have shown the heritability of cervical cancer is between 22 to 64%. The human leukocyte antigen alleles, HLA, and a combination of immunoglobulin-like receptors in association with HPV lead to HPV-related precancers. Genes associated with DNA damage and repair, ribosomal dysfunction, mitochondrial processes, and or oxidative stress have also shown to be associated with HPV persistence and cancer. With regard to hepatitis B, we know that older adults are at increased risk of developing chronic hepatitis B infection. Being overweight or having diabetes, we know increases the risk of having fatty liver, which increases the rate of liver disease progression in people with hepatitis B. Those who are on chemotherapy, steroids, or medications for autoimmune conditions are also at increased risk for hepatitis B reactivation, and people living with HIV, as well as those who are receiving hemodialysis, are, of course, at increased risk for hepatitis B because of exposure to blood and body fluids. Some of the genetic factors that lead to the impact of hepatitis B come from polymorphisms in HLA loci, variations in other genes that are listed here. Haplogroup F is a risk factor for chronic hepatitis B virus infection, and genetic polymorphisms of inflammatory cytokines such as TNF-alpha and IL-10 are linked to the progression of chronic hepatitis B infection. Turning now to precision public health, what is that? It's been defined as using better and more precise data to target disease prevention and control, and to improve health and health equity worldwide. Simply put, it is about implementing the right intervention at the right time, every time, to the right population. And we have certain technologies that Commissioner Califf was referring to that have helped us to enhance precision in public health. And these have really moved from the realm of research more into public health practice even. And I'll talk a little bit more about that here in a minute. But we think about the use of pathogen genomics that allow us to trace outbreaks, enhance surveillance and the use of big data and bioinformatics that allow us to analyze and interpret data in a very different way than we've been able to do in the past. And what that then allows us to do is to implement targeted interventions to disrupt these outbreaks and to understand and impact the infectious diseases that result in these outbreaks. So let's begin with pathogen genomics. And these are some examples of how pathogen genomics have helped us in the field of public health. Begin with the identification of a point source introduction of Vibrio cholerae from Nepal into Haiti, followed by clonal expansion that was attributed to the UN peacekeeping base there a few years ago. There was also an outbreak of listeriosis that occurred in 5 countries in Europe, where there were 47 cases and 9 deaths caused by this bacterium between 2015 and 2018. And it was possible to identify this clonal outbreak thanks to pathogen genomics. As you know, there's been an international hunt for the source of SARS-CoV-2, which is still ongoing. But much of that work is actually based on pathogen genomics. There was an outbreak of Mycobacterium camara that occurred a few years ago due to contamination at the heating, cooling unit devices manufacturing site that was able to be identified as the point source thanks to pathogen genomics. And finally, we are beginning to understand the dynamics of antimicrobial resistance in Mycobacterium tuberculosis transmission, thanks to pathogen genomics. With regard to surveillance enhancements and bioinformatics, the applications of digital surveillance using big data, mobility tracking, proximity monitoring, and what is called info-epidemiology has really disrupted this field entirely. One of the major ways that we've been using this technology is through wastewater surveillance. And this is sort of the bellwether that tells us, especially with COVID, has been able to predict increases in cases before they actually occur, as we start to see increases in the SARS-CoV-2 virus in wastewater. The application of electronic health records over the past couple of decades has also helped us to link health outcomes to public health issues that are ongoing. And smart or network technology, connection to Wi-Fi, GPS tracking, then allows us to actually track individuals and be able to determine how their movement leads to the spread of infections. So this is enhanced surveillance as well. And then finally, surveillance related to immunization. We have been able to do much better surveillance as a result of the application of bioinformatics to this field. Tempered with that are concerns about privacy and the potential for stigma to apply when it is discovered that an individual or a group of people may have been the source of a particular outbreak that might have occurred. And there is great promise, I think, in the future, including the use of artificial intelligence-driven modeling that might be coming down the pike for us. These two methodologies, including pathogen genomics and the use of bioinformatics, can then lead to targeted interventions, as I mentioned, where the older technologies of traditional microbiology are being replaced now with sequencing. We can do faster and better public health investigations and respond much more quickly to infectious disease outbreaks. And I've listed some examples of targeted interventions here where we are using these methodologies to enhance the development of better influenza vaccines, improving contact investigations where clusters of HIV infections occur, as I mentioned, detecting a number of different infectious diseases, combating antimicrobial resistance, again, Commissioner Califf referred to this as something that I'm sure you all will be talking more in depth about later today. Improving the health of subpopulations that might be in need of recommended prevention measures, and finally addressing and reducing health disparities that we unfortunately continue to see in our communities. So we know that there is hesitancy around many different vaccines, which has sort of been accelerated by the COVID vaccines. It's important to keep in mind that traditionally we have had this one vaccine fits all approach, where the same vaccine is used across the world in all different populations and in many different age groups. We know that some people do not respond to vaccines. For example, it's estimated that about 10% of people do not respond to the COVID vaccines. With hepatitis B, about 5% of vaccinees do not respond, do not make an immune response to hepatitis B vaccines. I'm one of those 5% of people. And I just presumed for the longest time that because I was vaccinated in my 30s, that was perhaps the reason that I did not make a response to the vaccine. But now we understand that perhaps it has something to do with my genetic makeup. And that is the reason that I did not make that response. So it's been known that there is a variability in host immune responses that's based on age, racial, ethnic background, and underlying medical conditions. And so what this has led to is the notion of what is called vaccinomics. This is a term coined by Dr. Greg Poland and their colleagues. They published a paper in 2007. And the idea behind this is very similar to what we have been using for a few years now in the therapeutic world of designing interventions, treatments that are specific to an individual's genetic makeup and their lifestyle and their the environment in which they live. The definition of vaccinomics, aims to understand genomic and systems level data to elucidate the basis of inter-individual variations in immune responses. And what vaccinomics has spun off, and this is really in its infancy, is this field now being described as adversomics. So it's a subset of vaccinomics looking at adverse reactions to vaccines. And here again, we know that there is significant variability in adverse reactions that people experience. For example, with the COVID vaccines, there are people like my husband who have had absolutely no reaction to the vaccine at all, no local reactions, no systemic reactions. And then there are others who are laid low for a couple of days because they have serious systemic reactions such as fever, malaise, etc., after a dose of COVID vaccine. And so I think the field is rife to understand why there is this wide range of differences between adverse reactions among different people from these vaccines. So I believe that advances in these two omics, vaccinomics and adversomics, can actually improve vaccine development and delivery and actually lead to safer and more effective vaccines in the future. Now, there have been noted genetic associations with the immunogenicity of vaccines, and this work has actually been going on for several years now. There are, in fact, 10 vaccines that are listed on this slide, on which a fair amount of work has been done, and I'm going to go through these fairly rapidly with you, but just to help understand the complexity of the outcomes with regard to immunogenicity that genetic background might have. And again, it's really dependent on a number of different genetic markers. You see some of these cartoon characters looking at different parts of the elephant, and it's kind of like that when we are looking at the impact of genetics on vaccines. The vaccine that's been studied the most is hepatitis B. And as I mentioned before, it's estimated somewhere between 4 to 10% of healthy individuals actually don't make adequate response, and about 5% actually don't make any response at all to hepatitis B vaccine. It's interesting that there is a high level of heritability, 77%, as determined in 207 twin pairs that were studied. I was talking to my daughter about this. She's a pediatric resident in Iowa City, and she said, oh, that's probably why I didn't make a response to hepatitis B vaccine either, because I was telling her about this talk and about how I had not made a response to Hep B vaccine. Turns out she didn't either. She had to have 2 rounds of vaccinations. The differences in immune responses to hepatitis B vaccine involve individual genes, allotypes, extended haplotypes, or single nucleotide polymorphisms. And the interaction of multiple genetic factors is actually what is likely to influence the immune response. Now, this is very, very complex, and I'm told you I'm going to go through it fairly quickly. But differences in immunogenicity to hepatitis B vaccine were actually noted to be associated with 277 genetic determinants across 117 genes. And HLA alleles accounted for 174 of these determinants. You see the specific HLA alleles that were negatively associated with immunogenicity. But interestingly enough, several HLA alleles were actually positively associated with immunogenicity. And null alleles of the complement component C4a gene due to deletion or non-expression were also associated with poor response. Toll-like receptors and cytokines and their receptors have also been identified as potential vaccinomics markers. Turning to COVID-19, if we take the systems biology approach, we know that race and ethnicity have an impact on the response to these vaccines, the immune state of the individual, certain genetic traits, as well as health status. So all of these are, in fact, involved in responses to COVID-19. So unfortunately, we don't have personalized vaccination at this point in time. So we're still going with the one-size-fits-all vaccination that can result in the risk of adverse reactions, as I spoke about, that varies tremendously. And it can also lead to short-term protection for certain individuals with a risk of reinfection. And in others, much better responses that can actually prevent disease. In people in whom the protection is short-term and not as robust, there is, of course, decreased effectiveness in preventing spread of the virus. What would personalized vaccination for COVID-19 do for us using the multi-omics biotechnologies that Commissioner Califf referred to? We would be able to better profile benefits and risks. So, for example, people who were recovering from a previous infection might not need to be vaccinated, or people who hadn't responded to a particular vaccine, we know it would be no use continuing to give them that same vaccine. They were non-responders, so we wouldn't give it to them. But for people who did respond to a particular vaccine, we would know, and we would be able to continue to give them the vaccine as needed. And we would also, of course, know about those who had severe side effects and maybe switch vaccines if we needed to for those folks. For influenza vaccines, differences in immunogenicity have been associated with 48 genetic determinants across 34 genes, with HLA alleles accounting for 20 of these determinants. The list that you see here shows those that were positively associated with immunogenicity. And then the third, the last bullet shows you the alleles that were negatively associated with immunogenicity. For MMR vaccines, numerous HLA and HLA genetic factors that individually or jointly contribute to the variability seen in humoral responses have been identified. With the measles vaccine, differences in vaccine immunogenicity were associated with 291 genetic determinants across 118 genes, with HLA alleles accounting for 60 of them. For the mumps portion of the vaccine, differences in vaccine immunogenicity were associated with 29 genetic determinants across 10 genes. And for the rubella vaccine, differences in vaccine immunogenicity were associated with 311 genetic determinants across 110 genes, with HLA alleles accounting for 84 of them. The diphtheria pertussis tetanus vaccine, differences in vaccine immunogenicity were associated with 4 genetic determinants across 3 genes for diphtheria, 5 genetic determinants across 4 genes for tetanus, and 12 genetic determinants across 10 genes for pertussis. Notably, most of these determinants were positively associated with immunogenicity. Turning to the Haemophilus influenzae type B, meningococcal and pneumococcal vaccines, differences in vaccine immunogenicity were associated with 3 genetic determinants for Hib, 13 determinants across 5 genes for meningococcal, and 25 determinants across 14 genes for pneumococcal vaccines. Notably, all 3 associations with Hib were negative, and all but 1 association with pneumococcal were actually positive. About half of the associations with meningococcal vaccines were negative. Polio and rotavirus vaccine, differences in vaccine immunogenicity were associated with 4 genetic determinants across 3 genes for polio and 2 determinants across 2 genes, along with Lewis blood group and secretor status for rotavirus. All but 1 of these determinants were positively associated with immunogenicity. So one of the things I mentioned earlier and Commissioner Califf talked about is misinformation and the hesitancy that has come about, particularly for vaccines, resulting from this misinformation. Part of it has to do with the adverse reactions that people experience receiving these vaccines. So the science of adversomics, as I mentioned earlier, it's in its infancy, but it is evolving. And as we talked about, vaccinomics primarily focuses on vaccine effectiveness, focused on immunogenicity, and adversomics deals with these adverse effects. Most of the studies that have been done have been for smallpox and MMR vaccines. And the reason I think that this aspect of the omics science is really important is that it has the potential for resolving public fears and concerns about vaccine safety. And I just want to point out, for those of you who are not vaccinologists or in the vaccine world, 3 notable cases of vaccine retractions. We've actually had to take vaccines off the market, or we've had to change our recommendations about vaccines due to side effects that were revealed in the post-licensure phase. Goes back to rotavirus vaccine from Wyatt Letterly at the time, called RotaShield, which in 1999 was removed from the market for causing a rare complication of intussusception, the bowel obstruction in babies that had received this vaccine. When the combination MMR and varicella vaccine became available, there was much excitement because you could reduce the number of injections given to children, but then we realized that this resulted in a greater risk of febrile seizures, and that might actually result in problems with children having further investigations done, emergency room visits, hospitalizations, et cetera. And so the preferred recommendation for MMR vaccine combined with varicella vaccine was actually withdrawn by the ACIP. And these 2 vaccines are recommended to be given separately for this reason. And then, interestingly enough, after the H1N1 pandemic vaccine became available, several countries in in Europe were noted to have a steep increase in narcolepsy, a rare disorder that happened following this vaccination, and probably has something to do with the genetic makeup of the population in those Northern European countries. A little bit about adverse omic studies for certain specific vaccines. We'll start with the hepatitis B vaccine. These studies have been less common, but more of a speculative nature, I would say. It's known that non-responders who carry the haplotypes listed here might be at greater risk of autoimmune disorders following vaccination. And certain HLA-DRB1 alleles that are listed in the next bullet point can result in activation of CD8 positive T cells, which can result in the production of high levels of interferon gamma and TNF and promote these autoimmune processes. Some recent whole genome transcriptomic studies have shown that hepatitis B vaccine non-responders had higher pre-vaccination expression levels of pro-inflammatory genes. I mentioned that there have been studies done in with the MMR vaccine, but they are also relatively uncommon. There are 2 SNPs in 2 genes that have been associated with MMR-related febrile seizures that are listed in bullet point number 2. There are case reports that have described mutations in genes in the interferon networks as a cause of severe adverse reactions to MMR vaccine, and those are listed in the next bullet point. There have been well-described clinical cases of exacerbations of autoimmune or rheumatologic conditions after HPV vaccination, particularly systemic lupus erythematosus, or SLE, in young women. This highlights the importance of the genetic predisposition of the individual, with perhaps an environmental factor acting as a second hit to provoke autoimmune diseases. And finally, GWAS studies have identified 2 nonsense mutations in cases of fibromyalgia following HPV vaccination. These have been associated with high levels of cytokines involved in inflammation exacerbation. So, getting to how can this knowledge, these technologies help us in rebuilding trust in government agencies, healthcare, and science through the application of precision medicine. So I want to start by saying that we will need to be patient, and this will take time. Unfortunately, we exist at this time in an environment where misinformation and disinformation drives a lot of the concerns that the population at large has, and has built this mistrust in governmental agencies, in healthcare professionals, in science as a whole. So we need to redouble our efforts. We need to work very hard and stay focused on effective education and communication with the public at large. Commissioner Califf talked about how important it is for us to speak in plain language to people, help them understand what we do, why we do it, and how it impacts them. I think the message as well as the messenger are really important. And while we may be experts in our fields, sometimes there are actually more effective communicators, such as community leaders, faith leaders, who are trusted and respected by the communities that we're trying to reach. We want to be careful as we move forward with these very powerful technologies of protecting privacy and reducing stigma that I mentioned earlier might be associated with some of the identifications we can make using these technologies. We are in election season, and it's very difficult to talk about this, but I do believe that it's really important that we depoliticize the field of medicine, of healthcare, of science, because it doesn't really seem to help when politicians enter into these discussions with limited knowledge, and really what they're offering is their own perspectives. And the public, of course, does not necessarily recognize that and might blindly follow what a particular politician might say. And that is not necessarily the best way to for them to gain knowledge and understanding for their own health. I think a multi-omics approach to vaccination is a very exciting thing to think about. It would be really important for us to understand who is more likely to benefit from vaccination and identify those who could develop adverse effects after vaccination, try to tailor vaccines to individuals, if you will. So I believe the future of personalized medicine is very bright, and I think it will probably continue to evolve more rapidly, perhaps, than some of us might imagine. I want to give you a quick summary of my remarks. I believe that precision medicine can improve infectious disease diagnostics, therapeutics, and prevention strategies. Precision public health is an emerging field where harnessing the power of pathogen genomics, enhanced surveillance using big data and bioinformatics with targeted interventions to rapidly identify and control emerging outbreaks would improve the health of all people. That vaccinomics and adversomics offer the promise of safer and more effective vaccines for individuals and the population at large in the future. And just a word of caution that while it's exciting to think about these new technologies, these approaches also need to be cost-effective and affordable. And the hope is that they will help us to restore public trust. Here are some of the references that I have utilized for my presentation today. I want to take a moment to thank my family. This is my husband, Samar, and my daughter, Maya, in the top left-hand corner. They are the pride and joy of my life. And to the bottom right is a photograph from a few years ago of my team at Chicago Medical School that allows me to do the work that I do in training the next generation of physicians. Thank you very much for your attention, and I'm happy to take any questions or comments at this point in time.
Thank you, Dr. Chatterjee, for an excellent keynote presentation. We have time for one question, and then we're going to take a short break before the next session. And for online attendees, if you have questions, please put them in the A&E Zoom box, and hopefully we'll be able to follow up with Dr. Chatterjee afterwards. If you can state your name and your affiliation, that would be great. Thank you.
That was a great talk, Dr. Chatterjee. That was a great talk, Dr. Chatterjee. I have a question or a comment, if you could shed some insight into which I am not aware of on the context of education. I feel that there's a gap education in general about vaccination, but also globally. I don't understand, for example, if the pneumococcal vaccine that's available here in the U.S., would somebody who is taking the vaccine in Asia or in Africa be exposed to similar kind of strains, so the vaccination will be effective or not? Especially because every few years the vaccines do change. Is it globally that these vaccines are effective or not?
Excellent question. So with regard to pneumococcal vaccines, you're absolutely correct that the serotypes that circulate in different parts of the world are different. And so the vaccines actually are different depending on where you are in the world. But I would say it's still a fairly blunt instrument, right? Because you're still using the same vaccine for sometimes millions of people. And we know that their genetic makeup, their lifestyles, their environments might be very different. And in fact, the response to the vaccines might be very different because of that. You probably also know that every year there is a new influenza vaccine, and some of you probably know that there are two different versions of the influenza vaccines every year. We license one for the Northern Hemisphere and one for the Southern Hemisphere based on circulating strains of the influenza viruses. But there again, you're talking about one vaccine that is for an entire hemisphere. And you can see where, again, that might be viewed as somewhat of a blunt instrument.
Thank you. Do we have any other in-person questions? Okay. Thank you, Dr. Chatterjee, for an excellent keynote presentation again, and thank you for joining us today. We're just going to have a 5-minute break, just a restroom break, and then we're going to return for 10:15 for the start of session 1. Thank you, everyone.
Thank you very much. Have a good day.
Thank you.
Bye-bye.
I'd just like to highlight that all the speakers in this session are virtual. So please hold your questions till the end of the session. And for our virtual attendees, please add your questions to the Q&A box in Zoom. So I would like to introduce the first speaker of our session, Dr. Stefan Kemp, who is a full professor in inherited neurometabolic disease and newborn screening at Amsterdam University Medical Center, University of Amsterdam. And please go ahead, Dr. Kemp, and share your slides while I continue with the introduction. Dr. Kemp specializes in X-linked adrenoleukodystrophy, or ALD, and inherited neurometabolic diseases. With over 25 years of experience, he has contributed extensively to ALD research with more than 120 publications and book chapters and more than 12,000 citations. Dr. Kemp also led the development of a boys-only screening algorithm for ALD as part of the Dutch newborn screening program, leading to nationwide screening from October 1st last year. His research group collaborates with ALD physicians and researchers worldwide, emphasizing open communication and collaboration. Dr. Kemp founded and is editor of the Open Access Worldwide ABCD1 Variant Registry and Disease Information Platform for ALD, a comprehensive resource in 5 languages with over 200,000 visitors per year for physicians and patients. He is actively involved in patient organizations serving on medical and scientific boards in Europe and the USA, and as a board member of ALD Connect USA. Dr. Kemp's Dr. Kemp's research team focuses on translational research, bridging basic science and clinical investigation, excelling in transformative publications that have shaped and continue to shape the field of ALD. Dr. Kemp, I'd like to turn it over to you now, and thank you for joining us today.
Thank you very much, Paula. Can you see my slides? I think so.
Yes, we can.
Super. Well, thank you very much for the invitation to present our research on adrenoleukodystrophy. So we'll start off, with my disclosures and funding. I'm an unpaid part of medical and scientific advisory boards, as Paula mentioned. And the work I'm going to present here from the PRISM ALD study is supported by grants from the Dutch Health Organization, ALD Connect, and the Virginia B. Truman Foundations, and the Dutch MetaKids Organization for Children with Metabolic Diseases. So we're going to talk about adrenoleukodystrophy, which is an inborn error of metabolism. It's a rare disease, but it has an incidence or prevalence of 1 in 15,000. And if you do a little calculation, even though it's a rare disease, still every year 350 new patients with this condition are born. The disease is caused by mutations in the ABCD1 gene, which is located on the X chromosome, and it affects the brain, the spinal cord, the adrenal cortex, and the testes. Just 2 slides on the genetics and the biochemistry of ALD. It's a fatty acid storage disease, and here we see a cell And fatty acids are produced through chain elongation for long-chain fatty acids. And in order to maintain the balance between fatty acids degradation and synthesis, these fatty acids need to be degraded in the peroxisome. And they are transported by the ABCD1 transporter. In case of a mutation in the ABCD1 gene, this transporter is defective, and there's a buildup of these very long chain fatty acids in the cell and in tissues. And while the genetics and the biochemistry are fairly simple because it's a monogenic disease, the clinical outcome or the clinical spectrum of patients is very unpredictable. So we can diagnose individuals at birth by demonstrating the accumulation of these very long chain fatty acids and demonstration of a pathogenic variant in the ABCD1 gene, but we cannot predict the clinical outcome of individual patients. So there are three core syndromes that patients can develop. And I will go into these in a little bit more detail in the next slides, but basically males are at risk of developing adrenal insufficiency. And the youngest patients that we have seen so far are a few months of age. Males are also at risk of developing a demyelinating brain disease, a leukodystrophy, which can start after the age of 3 years. And in adulthood, both males and females are at risk of developing a slowly progressive spinal cord disease, a myelopathy. The adrenal insufficiency affects about 50% of male patients before the age of 10 years. It's a treatable condition, but it's characterized by cortisol deficiency, which can be life-threatening. And what we know is that this is often not diagnosed initially. And what often happens is that patients are cannot in the hospital before they are diagnosed with adrenal insufficiency and subsequently with ALD. About 30% of the male patients develop the cerebral ALD before the age of 10 years. Here we see an MRI from a boy at the age of 7, 8, and 9 years, and you can see that this is rapidly progressive. Even though this is the most severe form of the disease, it's treatable, but it's only treatable when patients have not yet developed neurological symptoms. And if we diagnose these boys before the onset of neurological symptoms, they can be treated effectively with a bone marrow transplant. In adulthood, patients, males and females, can develop a slowly progressive spinal cord disease. For this form of the disease, there's no treatment. There's only disease-modifying therapy and supportive therapy available. ALD is becoming included in newborn screening programs in more and more countries. And the rationale for ALD newborn screening is shown on this slide. Here we see the three core domains: adrenal insufficiency in blue, cerebral ALD in red, and spinal cord disease in green, and the percentage of affected patients. And if we just zoom into the age of 10 years, we see that 1 out of 2 boys will develop adrenal disease, which is treatable, and 1 out of 3 boys will develop the leukodystrophy, which is also treatable if you diagnose these boys in time. So if we would diagnose the boys at birth, we could follow them up with and monitor them and initiate treatment in time. So this was already recognized in 2004 by my former mentor, Professor Hugo Moser from the Kennedy Krieger Institute in Baltimore, who at a nationwide newborn screening meeting mentioned that ALD newborn screening would save lives. Well, everybody agreed it was not possible at that time because there was no biomarker available for high throughput screening in newborn blood spot. The team at Kennedy Krieger set out to find this biomarker. And in 2017, the team reported C26 lysophosphatidyl-choline, or C26 lyso-PC, which is elevated in newborn UPRIC blood. It took another several years before the state of New York started newborn screening, and that we have to thank Elisa Seeger for this. She lost her son, Aiden Seeger, to ALD because he was diagnosed too late. And once she found out that it was technically possible to diagnose ALD at birth, she made it her life work to get ALD on the newborn screening program throughout the United States and, of course, in many other countries. So in 2013, Aiden's law was passed, and New York State was the first state in worldwide to start newborn screening. And Joe Orsini is the head of the lab in New York State who made this happen. And I can tell you from my own experience that this is a really, really difficult task. And in 2016, ALD was added to the recommended uniform screening panel list in the United States. Currently, 44 states in the US are screening for ALD, and that means that every year that 93% of newborns are currently screened for ALD. Outside the US, Taiwan started screening for ALD in 2016. And in 2021, regional pilots started in Israel, in Italy, and in Japan. And in 2022, a regional pilot started in Spain. And following a regional pilot in the Netherlands, last year, the Netherlands started nationwide screening for ALD, newborn screening. So currently, all newborns that are identified to newborn screening, I mean boys, they enter in a long-term, very invasive follow-up protocol. It means that from the age of 2 months onward, these boys are tested for their adrenal function, and this continues every 6 months. And from the age of 3 years on, 2 years on, every 6 months, these boys are receiving a brain MRI. And this, of course, is very, very stressful for the family, and I think also for the physicians, because we cannot predict the onset of leukodystrophy. So if we would be able to predict that, we would be able to do a risk stratification, and this will also likely allow us to initiate treatments and make better management decisions. But so far we cannot, as I said, we cannot predict the clinical outcome. So this is one of the main challenges that the field is facing in trying to address. So we really want a predictive biomarker. And yeah, well, this is kind of interesting because the definition of a biomarker actually came from the FDA, which says that a biomarker is a characteristic that is measured as an indicator of normal biological processes, pathogenic processes, or biological response to an exposure or an intervention, including therapeutic interventions. So there are basically 7 biomarkers that you can think of. A diagnostic biomarker to identify the presence or subtype of a disease, a predictive biomarker for the likelihood that the patient responds to a treatment or an intervention, prognostic biomarkers for the likely course of a disease or a health condition, and then there are 4 other categories of biomarkers. So in ALD, we're really interested in identifying either a predictive or a prognostic biomarker. We would like to have a prognostic biomarker for the leukodystrophy. So as I said, for the risk stratification, so that we can have boys of high risk or a low risk of developing the leukodystrophy. Because now they have to undergo an MRI twice a year, but also a prognostic marker for to follow the progression of the myelopathy, because we also know now that there are fast and slowly progressive patients. And the disease, we need a disease monitoring biomarker that correlates with disease severity for clinical trials. So the third thing you could think of is genetics, but already from the gene was identified in 1991, but soon after it became clear that the genetics do not have any predictive value for clinical outcome, because several monozygotic twins have been described in which only one boy was affected by cerebral ALD. And this is a study from our colleagues in Vienna, who describe a large family with 5 males, and all these males have a different clinical outcome. So it's clear that these genetics do not have any predictive value. Since 1999, I'm responsible or editing the ABCD1 variant registry. And so far we have cataloged over 1,200 different pathogenic variants in ABCD1, and it's clear that these mutations have no predictive value. And also the biochemistry. So in addition to the genetics, the biochemistry, we do know that all ALD men have elevated very long chain fatty acid levels. And the total plasma very long chain fatty acids, or in blood cells or in fibroblasts, they do not correlate with the phenotype. So this is again the 3 core syndromes of ALD: the adrenal insufficiency, the myelopathy, and the cerebral ALD. And the current hypothesis is that the default mode of ALD in males is the development of adrenal insufficiency and the spinal cord disease, and at the onset of the cerebral ALD is, for that there are modifying factors, which can either be genetic variants in, or maybe rare genetic variants in modifier genes, but also environmental factors. Boys have been described that following a head trauma develop cerebral ALD. And also viral infections are suspected, but have not been demonstrated any virus infection that could cause a cerebral ALD. Many biomarkers have been studied in ALD, and I have just one slide here that summarizes these. Glutathione in plasma is reduced in all ALD patients. MCP1 in cerebrospinal fluid, or total protein in CSF, or chitotriosidase in CSF is all increased in cerebral ALD. And antibodies against profilin-1 are in plasma are increased in cerebral ALD. Superoxide dismutase, or SOD, is highest in controls, lower in spinal cord disease, and lowest in cerebral ALD. And neurofilament light chain in plasma, but also in CSF, is high in cerebral ALD, higher than in non-cerebral ALD, and that's higher than in controls. So these were promising studies, but subsequent studies have shown that almost all of these studies were either underpowered, they were not reproducible, they showed limited association, or they identified biomarkers that are not predictive. For example, NFL is high in patients with cerebral ALD, but it becomes high at the time they develop cerebral ALD. So it's not a predictive biomarker. There's one other study that I would spend one slide on, that is a SNP in CYP4F2. CYP4F2 is an interesting protein because it also is involved in omega oxidation of very long chain fatty acids. That means that a very long chain fatty acids, saturated very long chain fatty acids, is converted into a dicarboxylic acid, and these dicarboxylic acids, they have an alternative entry into the peroxisomes. So this is an escape route, but it's it has a very low activity in cells. Only like 5 to 10% of all fatty acids go through this route. We identified a SNP in CYP4F2, valine at position 433 to methionine, and what we could demonstrate at this SNP results in a lower amount of protein, and this also correlates with a lower amount of activity of this protein. So while this was very interesting, and we could demonstrate that the risk that this SNP is associated with an approximately 2-fold increase in the chance of developing cerebral ALD, this was not predictive. So we need to do better, and the difficulty in ALD is that patients may accumulate symptoms over time. So a patient that has adrenal insufficiency at this time may still develop cerebral ALD or myelopathy in adulthood. And also patients that have developed myelopathy in adulthood are still at risk of developing cerebral ALD. So in order to have very strong and good clinical cohorts, you need to initiate a prospective long-term studies, and these with regular sampling. And these biosamples must be collected and stored in a standardized manner, and they must be linked to detailed and accurate clinical information of these individual patients at the time of sampling. And the problem, of course, is for many rare diseases that ideally you would have large international cohorts and biobanks, but establishing these are very challenging due to the complex regulatory issues and sample ownership. An alternative, of course, could be that different groups working on a similar disease follow standardized follow-up protocols across the cohort, and to ensure data compatibility, and also have standardized protocols for taking biosamples and maintaining these. In Amsterdam, at the Amsterdam UMC, my colleague Mark Engelen started the Dutch ALD cohort over 10 years ago, and currently there are 120 ALD patients enrolled, and they visit the clinic every year, and they participate in a lot of clinical trials and clinometric studies, MRI studies, but also every year they donate biosamples, and these are collected in the biobank. And this is a collaboration between the Amsterdam Leukodystrophy Center and the Laboratory of Genetic Metabolic Diseases that performs a lot of diagnostic research for peroxisomal diseases and mitochondrial diseases. So what we set out to do is with the aim to identify prognostic biomarker is have the whole cohorts every year visit the clinic, do deep phenotyping, and take biosamples. These biosamples we use for various studies: lipidomics, proteomics, whole exome genome sequencing, RNA-seq. Of course, it generates an enormous amount of data, and for that we collaborate with our colleague Martijn Schut, who's the professor in translational AI, to make some sense out of all this data. And not a topic of this presentation, but what we also do, we collaborate with Vivi Heine of the Stem Cell Laboratory to make iPSC cells, astrocytes, organoids from these patients, with the same idea to get more insight into prognostic biomarkers. This is a laboratory in Amsterdam. This is both a diagnostic facility and a part of it is a research facility. And I would like to spend a few slides on the presentation that was published 2 days ago in Communication Medicine, which is the first product of this work. So we did deep phenotyping to generate different cohorts of phenotype patients, and we performed lipidomics. This generated a lot of potential new biomarkers, all lipids that have very long chain fatty acids in the side chain as fatty acids attached to it, both for males and females. And interestingly enough, if we look at C26 lyso-Pc, which is also the diagnostic marker as well as the newborn screening marker, in contrast to what was published in the past, looking at plasma variant chain fatty acids, if we look at lyso-Pc C26 in these well phenotyped and well characterized patients, we see that in patients that have cerebral ALD, this marker is much higher than in patients that have no cerebral ALD, but only mild spinal cord involvement and are past the age of 55 years. We should also see a correlation in patients that have developed spinal cord disease, which is either fast progressive or slowly progressive. Also in patients that have adrenal insufficiency or no adrenal insufficiency. And also in women, we see a correlation with disease severity. And these 2 yellow dots here are 2 females that have developed cerebral ALD, which is very rare in women. But in these 2 women, which have the highest level of variant chain fatty acids or C26 lyso-Pc, these have developed cerebral ALD. A summary, just a summary of the data that we have produced, which is important for follow-up in newborn screening. is shown on this slide. We have patients that have both cerebral LD and adrenal insufficiency, and these the dots in red, and we see the C26 lyso-Gb levels. And then this is the most severe form of the disease, and the mildest form of the disease are patients that are over the age of 55 years, have no adrenal involvement, adrenal insufficiency, and only a slowly progressive myelopathy. And we see that these are all clustered together here. And this is going towards a risk stratification, because it seems that there is a cutoff of around 400, and above there's a higher risk of C26 lyso-Gb. Of course, this is just one biomarker, so this is not the entire story. And that brings me to my last slide. ALD has an unpredictable clinical spectrum, and that complicates the identification of prospective biomarkers. The disease severity is determined by genetic, epigenetic, environmental factors. The deep phenotyping, which we do now, is the key to identifying predictive biomarkers. There's a strong association between plasma variant chain levels. There's a strong association between plasma levels of variant chain fatty acids containing lipids and disease severity. And high levels of C26 lyso-Gb correlate with risk for cerebral LD, adrenal insufficiency, and severe or fast progressive spinal cord disease in male ALD patients. What we're currently performing is a whole genome sequencing of the entire cohort, because now we have these well-defined groups, and we think that we now have a better chance to find modifiers in genes. And in the end, we expect that there will be a panel of core biomarkers, which can be genetic, lipids, and likely other factors that will improve the prognostic and diagnostic accuracy of ALD, which will enable risk assessments and precision medicine following newborn screening. Yeah, and a very important lesson is that in rare disease, it's really, really important to collaborate with colleagues, and because you need to do that to generate large patient cohorts. And with that, I come to my last slide. I acknowledge the people who are involved in this part of the research. And I have one more slide, and I like this analogy, because what we've learned over the years is that, like astronomers using the Bortle scale to reveal faint stars in dark skies, our deep phenotyping approach uncovered previously hidden correlations between variant chain fatty acid lipids and ALD severity, potentially revolutionizing disease prediction and treatments. And with that, I would like to thank you.
Thank you, Dr. Kemp, for that excellent presentation. We're going to hold questions till the end of the session. So next, I would like to introduce Dr. Dan Theodorescu from the Samuel Ocean Comprehensive Cancer Institute of the Cedars-Sinai Medical Center. Dr. Theodorescu, if you could go ahead and start sharing your slides, I'll continue with the introduction. Dr. Theodorescu has emerged as an international leader in translational cancer research. His pioneering application of computational biology led to the discovery of genes that drive bladder cancer and other cancers, while providing novel biomarkers and therapeutic targets, as well as the foundations for the personalized precision therapeutic approaches such as the molecular twin precision oncology platform and the coexon principle. He's also led the discovery and development of a first-in-class RAL GTPase inhibitor as a new therapeutic in several human cancer types. His lab was the first to demonstrate that the loss of the Y chromosome in cancer makes tumors more aggressive by allowing them to evade the immune system, and that this advantage becomes a disadvantage or a liability by increasing their sensitivity to immune checkpoint inhibitors, which has broad implications in cancer medicine, since many tumors lose the Y chromosome. Dr. Theodorescu has been elected to the American Society for Clinical Investigation, the Association of American Physicians, the American Surgical Association, and the National Academy of Medicine. He is an honorary fellow of the American Association for the Advancement of Science. I'll hand it off to you now, Dr. Theodorescu, and thank you for joining us today.
Okay, it's can you see the slides okay?
Yes.
Okay, great. Well, good morning. thank you very much for the kind invitation and the kind introduction. What I'd like to do today is describe one of our latest advances, which is the Molecular Twin Precision Oncology Platform, or MTPOP for short. This is a tool that we hope will lead us to better discoveries, development, and delivery, and ultimately democratization of precision medicine for cancer and perhaps other diseases. So these are my disclosures. And a number of comments have been made about precision medicine and personalized medicine. I'm going to show this slide because I wanted to sort of give you a lead in to the whole topic of multi-omics. And essentially, the terms precision medicine and personalized medicine have somewhat emerged in parallel with perhaps personalized medicine arising first. But in essence, let me put the pointer here. In essence, personalized medicine evolved first as an offshoot of the Human Genome Project. And the concepts were clear with the 4P models: predictive, preventable, personalized, and participatory. And then further definitions and refinements were made to describe it as reactive, moving reactive medicine to being a proactive discipline. But importantly, in 2013, the concept that it wasn't just going to be genomics, but that proteomics would be also involved. And in parallel, in the concept of precision medicine started emerging in about the 2010 or so in the literature, followed by a National Academies report defining, if you will, precision medicine and indicating that it's the preferred term because it conveys the concept that is more accurate for individuals. And this is going to be the sort of the theme of the reason of why we developed the molecular twin. But importantly as well, also made a point about the concept of multi-omics. So the concept of multi-omics is pretty old if you look at those two chronologies. And so it's been talked about a lot, and nevertheless, precision medicine could still, as a whole, if you will, as a broad statement, could still, at least in cancer, be advanced and refined, since we're not able to predict with great precision many of the diseases that we want, either their outcomes, natural history, or response to therapy. So what we basically hypothesized a couple of years ago is if we think holistically as, again, speaking in the context of cancer, but applying to potentially other diseases, if the outcome of a patient really ultimately is a dynamic between the interaction, in this case, of a tumor and the patient, that dynamic needs to be sorted out and studied simultaneously. So it's not sufficient just to do a biopsy and basically look at the cancer and then say, well, we can predict this, this, and this. That may work. That may offer some advantage, and it does in many cases, but not in all. And we're probably losing a lot of individualized predictive ability by not looking at the host. So our hypothesis was to look at not only a multi-omic approach, but actually look at a holistic one where we took a lot of other information regarding the host, including the omics from both the cancer and the patient. So with that in mind, this is really the architecture of the molecular, what we call the molecular twin. So we basically have a patient, and as you'll show you a bit later, we actually sample the host in a variety of ways. And again, when I think of samples, I think of samples of all types. So clinical history, those are elements that we integrate into the model. And of course, tumor is characterized in a variety of ways, both clinically and as well at the molecular level. We also take information from external databases, and I've shown here a couple, but there are more than that we use, and feed this in, including data from basic science, which is included, and of course the external databases, but specifically from our cancer center, and as you'll see why as again later in the talk. So with all that is brought in using machine learning, artificial intelligence, to create basically a predictive, depending on what questions we're asking, that will allow us to predict better disease outcomes, both treated and untreated, as well as the response to therapies. But importantly, will also be a platform for the discovery of new therapeutic targets, biomarkers, and ultimately be amenable, and this was a key criteria when I developed sort of the architecture concept, to be able to be democratized throughout the world. And again, ideally identify markers, and that would lead to novel therapies, both in the prediction and the prevention and the proactive sort of arenas, not just therapeutics. So what does this entail from a operational point of view? So the cancer patient comes to the clinic, and then we collate a number of different clinical information, as well as what I call sensor data, which includes a number of things, but the most obvious ones are imaging that the patient has, and then we take a number of samples, tissue, both normal and cancer. Again, it's about the host as well as the tumor. And I've shown here microbiome, but that includes as well other body fluids. And then for the images and clinical data, we use computational processing as well as NLP to pull out a lot of the key variables that are of import, and quantify them accurately for the AI engine. So that's, of course, more easily done with the molecular profiling. And then at the same time and in parallel, we generate models in a variety of ways, either using iPSC or xenografts, et cetera, to look at the host and the tumor simultaneously, hence the iPSC concept. And of course, this information, as I showed you in a previous slide, goes in with external databases as well as bench research in our cancer center and others into the AI engine. And then it creates what we call the molecular twin, which is important to remember that that molecular twin gets annotated and advanced and enriched and evolves basically as the patient goes through their therapeutic and follow-up journey. So we have repeated samples, and those samples get integrated in this molecular twin architecture, and if you will, wiring of the twin mechanically is altered as you go on. And then what we do with the molecular twin is two things. We use it for the present and for the future. So the present, of course, we do a lot of sequencing, DNA sequencing, and that is immediately actionable because we do it in a CLIA/CAP certified environment. And therefore, the clinicians have now the ability to look at the molecular twin report in our electronic medical record and basically select the best standard of care option for patients based on their molecular features of their cancer in cases where that is established standard, and importantly, match patients to clinical trials that require certain molecular characteristics. But what we really want to do, and I've begun doing for the future, is create a new discovery of new targets and biomarkers. So that will require doing molecular twins on many patients, collecting their samples, doing the twins, and then following them very carefully as their disease progresses or regresses. They do well, they don't do well. And for those, obviously, that don't do well, we basically hope to identify new drug targets and then develop new drugs. For those that do well, and of course, it's a two-sided coin, we need biomarkers that are prognostic, predictive, parsimonious, which I'll get to, and diverse. We have, as you'll see later on, we have a very large and very diverse and rich population here in Los Angeles County. We have a very great focus at our cancer center to look at this great diversity and enrich our biological knowledge based on what we find in this diverse population. So what have we done so far at Cedars Cancer Center with the molecular twin? We've enrolled about 2,500 patients, and we're enrolling about 1,000 to 1,500 patients a year. Importantly, these are patients that are enrolled with consents, IRB-approved consents, and we do this because we want to be able to do research with consent on their samples, and also because we want to recontact them, et cetera, and follow them lifelong. And we, again, as I mentioned, we're very attentive to our diversity and disparities in our catchment area, which is Los Angeles County. So this is what we've done so far. We've collected quite a few samples, 2,000 plus, and I show you this because these are our priority sites that we've started. We have now extended to a larger number of sites that are, again, priorities for our cancer center. But this is purely bandwidth driven. It's not science driven. We have interest in many of these tumors, but these are our priorities. And of course, we have a number of samples of a variety of types: blood, aliquots of stool, and of course, FFPE for the cancers. Importantly, speaking of diversity and demographics, you see here that we have a significant diversity. I realize that you may not be able to see the color codes, but suffice it to say, you can see that there are a lot of other colors beyond the main colors, and that indicates that we have a very, very significant diversity in race, ethnicity, and of course, gender and gendered cancers here based on their biology. So we have great opportunities again for discovery. So let me take you to a hypothetical now before I give you a concrete example. We've gone through this hypothetical of what, or the actual construction of the twin, and what the ultimate goal is here is, if you will, cartooned by these two boxes that are hopefully moving on your screen. And these two boxes show a bunch of dots, and those dots are molecular twins of patients that have been cured after specific treatments. Okay, so this is an abstract concept. You could take any treatment you want, say immunotherapy, for example. Let's just take that because the example is going to be immunotherapy in bladder cancer, which is the area that I focused on with my research lab. And so these are dots of patients that have responded, that have survived, and patients that unfortunately have died. So, okay, so patient X comes in, and basically here we are. We have patient, we do the molecular twin, and that molecular twin of that patient is now going to be mapped into the architecture, whether in one of these boxes. And this patient, it turns out to be lucky because they got mapped in the cured box. So the map suggests that they are in the teal cluster, and that teal cluster is one where they're cured after immunotherapy with PD-L1. after their diagnosis of metastatic bladder cancer. That is the dream of what the molecular twin can do. So let me now give you a concrete example how this all comes together. We picked pancreatic cancer for a couple of reasons. One is it's a priority of our cancer center. We have an incredibly vibrant multidisciplinary team and clinical activity. And as you could see from the slide, both in terms of the incidence and in terms of its progress, its incidence is significant. We haven't made much progress, as you can see on the right here, in terms of mortality trends, in terms of this horrible disease. So what is the standard of care of this disease right now? The patient gets staged, a biopsy is done in some cases, not in all, and then what's called a resectability classification is done, which is really the only potentially curative therapy of this disease, unfortunately, is one, which is taking it out, surgery, which is called the Whipple operation. It's a very, very big operation. It involves removing the pancreas, part of the duodenum, small bowels, reconstructing everything. It's a long operation. It has significant morbidity, as you can see here, and it has a significant mortality. But the unfortunate part of it is that even in the best selection criteria and best hands, surgically skilled surgeons, the 5-year survival is only 20% or so. So the resectability, borderline resectability, non-resectability is a key determinant. And clearly, what we're doing is we're resecting patients that actually are not curable, because these patients, when they do recur, they don't recur at the surgical site, they recur mostly metastatically. And the tools we have is imaging, CT scans, etc. We have CA19-9, which is a marker that's been around for some time, and I'll give you a little anecdote. I only have one cartoon. This 19-9 marker is established for this use, but this is the year it was developed, and some of us were in middle school. Some of us, I'm sure in the audience, were not even born yet, but this is the era of Grease and other cartoons, Star Wars. It's a long time ago. So changes and improvements are due. So here are the changes and improvements. So we applied the molecular twin concept first on this disease using samples that we had with appropriate consenting of patients. We had about 70 or so patients that we scored in terms of survival. And here, what this shows is really the methodology, which is quite sophisticated and extensive. I'm not going to go through it in detail in the slide, but essentially we take all the analytes, including the clinical, like I said, that's all quantified using various digital technologies, as well as the slides, as you'll see in a minute. We use a variety of AI and other technologies, mathematical technologies, to really look at the single omic predictors and the multi-omics, and then did leave one out cross-validation of the data set for the biomarkers. And what we're trying to predict here is really survival after the Whipple operation. So basically a better CA19-9. So, but the ultimate test was external validation, which we did in actually 4 different independent cohorts. So this is an example here of multi-omic, and the multi-omics I picked is one of image analysis and computational pathology. Again, a very extensive multi-step process. We first had to do the sampling, then we had to develop algorithms to really identify only the cancer cells. Pancreatic cancer is a cancer that is highly infiltrated by stromal cells. So the identification of the cancer and the stroma had to be done very carefully. And then a variety of algorithms were used to identify nuclear features and other architectural features that were then put in to mathematical models to come up with a feature visualization identifying the most powerful features of the computational, the image analysis of the cells. So these are very abstract concepts because the computers identify features of, say, the nucleus that have really no optical, sort of visual, recognizable identity by a human. So nevertheless, they serve as variables. Think of them as a RAS mutation. Same thing, same concept. Mathematically, they're treated the same way. So when you do this, and you do it for all the analytes that I showed you in the previous cartoons, you come up with a multi-omic model. And that multi-omic model, we are very pleased to see that even with very limited number of samples, we had an accuracy and a PPV, positive predictive value, that was very high. And here is the multi-omic in green, and then we have the single omics here in decreasing order of importance. You see the clinical and surgical pathologies right here in the middle. And the CA19-9 is alone really at the bottom. Interestingly, when you look at the model and the external data sets, and importantly in this external data set, because we wanted to move this towards the clinic, we only looked at what variables and information the physician would have before the Whipple in order to be able to make this decision. And you can see here that in that sense, the clinical and the plasma proteomics were very, very significant and good predictors, with the CA19-9 being a distant second. Terrible, really, a PPV. Not so terrible in the second cohort in terms of PPV, but again, clinical and plasma proteins were there. We also identified networks of potential druggable targets at the protein level and both the tissue protein and the plasma levels, and we're pursuing a number of these in the lab. Here is one of the more important slides, because what it does is it takes us to the parsimonious word, which I mentioned earlier, which is the road to democratization of this test. And what this shows is a graph of the various analyte compositions, and you may not be able to see what this is in the multi-omic model, but suffice it to say, these colors represent the various analytes. So pathology, DNA, RNA, plasma proteins, and you could see that as you reduce the number of features, which is the variables, basically this oscillates somewhat, but then it peaks at about 590, and then starts stabilizing and going down. If you look at the various other reduced mixtures with computational pathology, clinical, plasma, computational pathology, and clinical, you see that you're getting pretty decent numbers, even with the pre-surgical composition, which is plasma and clinical. And so if you look at that in more detail, you look at and see that about at 470 analytes of plasma, as well as computational pathology and clinical, we have some very good predictors with not a lot of variables. So what that leads us to think is potentially, can we build a point of care testing scenario that we reduce cost significantly? So we have a world-class proteomics facility at Cedars, and they have been at the cutting edge of a variety of technologies, and now are able to carry out biomarker assessments using a Demetra device, for example, on up to 500 or so proteins, which are more than enough to cover the ideal optimal for the previous graph I showed you. So what is the dream here that I have is potentially take the molecular twin concept using this democratization tool that the artificial intelligence is able to offer us to focus on the most important variables and potentially convert a tens of thousands of dollar precision medicine approach to something that is much more accessible across the entire world. So in the future, what I'm hoping to see is the molecular twin showing up here and making better predictions of who's resectable and who's not, and therefore operating ideally only in the 25% of patients that are going to do and be cured from the operation. That's the ultimate dream. We're validating the we're actually making the test in a clinical actionable way now in order to be able to test this on a much larger scale. And of course, some of the things we learned from the twin, we could use in adjuvant and surveillance approaches. So putting it all together, we'll see patients in cancer, but we're extending the twin to other diseases. And we're collecting these twins, we're characterizing them and using it today for standard of therapy use or not. They're cured. If they're not cured, they're eventually going to go into clinical trials at some point using molecular twin guided clinical trial selection using new therapeutics that are developed based on the molecular twin that hopefully will be commercialized in tests and drugs. And we're testing it, obviously, a number of them through our network of hospitals in our health system. But ultimately, we want to test it nationally and globally. The paper that I described on the pancreas was recently published. It came out in January of this year, and it's freely available to the scientific community. And the last slide is to thank my collaborators. Primarily, this was driven by the proteomics, our genomics infrastructure. And this is a special thanks to the team that was contributing to this, including genomics, proteomics, and the pancreas multidisciplinary group that were the subject matter experts in this adventure that created the first molecular twin prototype. So thank you very much for your attention. Appreciate it, the opportunity to share this with you.
Thank you, Dr. Theodorescu, for an excellent presentation. So next, I would like to introduce our final speaker, Dr. Douglas Fowler from the University of Washington. Please, Dr. Fowler, could you go ahead and share your slides while I do the introduction? Dr. Fowler is a professor of genome sciences, adjunct professor of bioengineering, and a member of the Brossman Beatty Institute for Precision Medicine. Since 2019, he has served as the director of the Center for the Multiplex Assessment of Phenotype, a National Human Genome Research Institute Center of Excellence in Genome Sciences. He co-directs the Center for Actionable Variant Analysis and is a founder and current executive committee member of the Atlas of Variant Effects Alliance. The Alliance consists of a diverse group of over 700 people, including experimentalists, computational biologists, clinicians, and others with the shared vision of determining the effect of every possible variant in every gene in the human genome. Dr. Fowler's work has focused on developing and implementing new technologies to address difficult problems in genomics. He is a global leader in high-throughput sequencing-based assays. His lab has extensive and deep expertise in large-scale experimental approaches to interpret the clinical consequences of human genetic variation. Dr. Fowler holds a bachelor's degree in chemistry cum laude with departmental honors from Northwestern University and a PhD in chemistry from the Scripps Research Institute. Dr. Fowler, turning it over to you, and thank you for joining us today.
Thanks so much for that kind introduction. It's really exciting to be able to share some of our work with you. My lab generally, as you said, works at the intersection of genomic technology, protein science, and increasingly clinical genetics. And so I'll tell you today about some of our efforts to try to understand the effect of human genetic variation, including in pharmacogenes, using new functional genomics tools that we've developed. So The idea for what we're up to proceeds from the notion that we have a great understanding of the grammar and coding regions of the genome, right? We know how DNA specifies a transcript and a transcript specifies a polypeptide sequence, but our understanding of what happens gets much fuzzier when we move to function, right? So how polypeptides fold up into functional or non-functional proteins, how those proteins specify cell types and cell functions, and how cell tissue and developmental phenomena roll up into disease are much more difficult to predict and understand. And as a consequence, when we find a variant in the genome, we may know how it will affect the polypeptide sequence of a protein, but it's much harder to predict and understand what the effect of that variant will be on the molecular, cellular, and organismal level. And so as a consequence of that lack of understanding, in the arena of genomics and clinical genetics, we're in a situation where we can discover genetic variants easily and are doing so at a very large clip, both because of research studies and clinical genetic testing. But we have trouble when we want to understand what these variants may do in a particular patient. And that's reflected here in an analysis of the ClinVar database, which is where clinical geneticists and genetic testing companies deposit the results of their analyses. And so what you can see is that about well over 80% of the variants in this database at the moment are variants of uncertain significance. And those variants of uncertain significance are accruing at a massively increased rate compared to more definitive interpretations like benign or pathogenic. And variants of uncertain significance arise when the clinician or testing lab doesn't have enough information about the variant to determine whether it is pathogenic or benign. And these variants represent a sort of dead end in clinical genetics and precision genetic medicine, because you can't use a variant of uncertain significance to make a diagnosis or devise or alter a treatment plan or decide on dosing a drug if the variant is in a pharmacogene where such genotype-guided dosing would be advisable. So in any case, this represents one of the fundamental problems in translating genomics into the clinic, and it's the problem that we are trying to solve. Additionally, it's worth noting these plots show you the variants that we've discovered so far. But if you do the math, every single nucleotide variant that's possible in the human genome, all 9 billion of them, will be present only in about 50 individuals alive today, meaning that as we sequence more and more people, we'll eventually find every variant compatible with life, but not that many times, which sort of illustrates the scale of the problem that we're trying to solve. So what kind of information can we collect about any variant in principle? Well, we can collect functional data based on an experiment, right? So for many decades, we've been able to make a variant protein or cell or model organism and compare the phenotype of that variant to a reference sequence. And in so doing, make a conclusion about what the variant does, like whether it alters the enzymatic activity of a protein or alters the behavior of a cell, and so on and so forth. And that information is very useful in the clinic, but it's hard to collect and often comes too late to help patients because traditional assays, one-at-a-time assays, don't scale. So while functional data is in principle a way to solve this problem, in the past, it hasn't been a practical solution because of scaling. And so what we've done, along with a host of other people in this field now, is devise a set of multiplexed assays of variant effect in which we can make all possible single nucleotide or missense variants in a protein of interest and test their effect en masse, reading out the results using high-throughput DNA sequencing. And the simplest type of multiplexed assay is a growth assay. That's maybe shown here in schematic format, where each cell in a pool expresses a different variant in your protein of interest, and you've rigged up the assay such that the cell growth is dependent on the function of the protein. So cells harboring wild-type-like variants grow happily and expand in the population, and cells harboring loss-of-function variants, die off in the population. So we can use high-throughput DNA sequencing at the beginning, middle, and the end of the outgrowth to quantify the frequency of each variant, and then look at how each variant's frequency changes in the pool and compute a functional score that tells you, relative to wild type, the ability of the variant to support growth in the assay, and thus its function. And you can organize that data into a sequence function map like the one shown here, where each column is a different position in the protein in the protein of interest. Each row is a different possible amino acid substitution, and the tile at each position and amino acid combination tells you the functional score from blue, meaning loss of function, through white reference-like function, and red enhanced function. And so collecting this type of data means that you know the function of every variant in the protein, all the ones you've seen before, including all the ones you'll see in the future. And we and others have elaborated all kinds of different assays that can be used in this multiplexed format, including many that you'll be familiar with and some that I'll talk about today, like for enzyme activity, protein ligand binding, protein abundance, as well as cell morphology and behavior, fluorescent reporter assays, and single cell RNA sequencing. And these types of assays have been developed in phage display bacteria, yeast, cultured human cells, and beyond. And so we also have shown that these data are incredibly useful at solving the variant of uncertain significance problem. So this are just some figures from a paper we published a number of years ago, where we collected multiplexed functional data, or others did, for 3 key genes in which germline variation can cause pathogenic variants in these genes, can cause a massively increased risk of cancer over a person's lifetime. And I'm sure you've heard of many, some of these genes at least, BRCA1, TP53, and PTEN. And what we did was partner with a genetic testing company, Ambry, and we went through and asked, how does having functional data alter the interpretation of the variants that Ambry had found over the years in these genes? And what we found was that using the functional data that had been generated could eliminate at least 15% of the VUS, the variants of uncertain significance, but in the case of PTEN, but for BRCA1 and TP53 could in fact eliminate more than half of the variants of uncertain significance. So this really illustrates how having this data can massively improve our ability to interpret variants. And in this study, each one of these reinterpreted variants of uncertain significance that became either a benign variant or a pathogenic variant, like that information was returned to the patient and their clinician. And in the case of these cancer risk syndromes, the patient was able to be offered, either risk-reducing surgery or enhanced surveillance for cancer. So this shows how we can use functional data generated at scale and use it to improve patient, to give a molecular diagnosis and then offer treatment options. So that's sort of where we are today. Where we want to be is to generate variant effect data for every gene that's disease-related in the genome, for every one of the phenotypes that we think will be relevant. And to do that, we are missing a bunch of technologies, and that's what my lab is currently working on. So we're missing technologies to scale. We're missing technologies to apply these types of multiplex assays to every protein encoded in the genome. We're missing technologies for some phenotypes, and we're missing technologies that allow us to test variant effects in the appropriate genetic, cell, tissue, and developmental and environmental context for the disease that we're interested in for that protein. I don't have time to talk about all of this work today, obviously, so I'm just going to pick 2 quick stories, one of which is focused on a set of pharmacogenes, and then I'll tell you a little bit about kind of our overall goal and how we'll get there, and that's how we'll spend the next 15 minutes or so. Okay. So let's talk about a type of protein that we didn't have a multiplexed assay for, but for which we built one, and that's secreted proteins. So I told you that to do a multiplexed assay, like a growth assay, you need the DNA that encodes the protein variant to be in the same cell as the variant protein. So you can use the trick of high-throughput DNA sequencing to track the variant frequency and generate a score. Well, that doesn't work for a secreted protein, right? Because if you make a library of cells expressing secreted proteins, then each one of those cells is going to turn out protein. It's going to be lost to the media, and you won't be able to link the protein variant to the DNA variant and use high-throughput DNA sequencing. So this is an important problem because about 10% of the genome encodes secreted proteins, and these proteins, many of these proteins are related to disease or are therapeutics themselves or both. And that's the case for the disease and protein that we chose to focus on to develop this technology, hemophilia B. Hemophilia B is a clotting disorder where patients are prone to bleeding, and missense variants, primarily missense variants in a gene called factor IX, which encodes the factor IX protein, disable this protein in some way and prevent the normal clotting cascade. And the therapy for this disease is to give recombinant factor IX. So factor IX is both the cause of the disease and the drug. And this is a rare disorder that's X-linked. So as with many other rare diseases, there are many variants of uncertain significance in this gene, and we would like to be able to give molecular diagnoses to these patients, but we can't because we don't have enough information. So we decided to develop a technology we call multiplexed surface display of extracellular proteins, or multi-step. And in this technology, what we do is use a cultured human cell line and engineer it so that we can express a variant of factor IX or any other secreted protein. And that variant, rather than being lost to the media, is attached to the surface of the cell using a short linker and a transmembrane domain. And so what we can then do is take our pool of cells, which are, each one is expressing a different variant of factor IX, but collectively they express all 10,000 or so missense variants of this protein. And we can stain the pool of cells for one of many antibodies or other affinity reagents that allow us to read out some aspect of the function of this protein. And so today I'll tell you just about a variety of antibodies that we use to either read out the presence or absence of different parts of the protein on the surface of cells, like this light chain antibody here or heavy chain antibody, just tell us, is this variant secreted or not? And then another antibody that binds specifically to a post-translational modification that is critical for the function of this protein, a gamma carboxylation of glutamic acids in a particular domain of the protein. And so this post-translational modification reminds me to tell you that we have to do these assays in cultured human cell lines, because this is a complicated protein that sustains many post-translational modifications, including protease cleavage and the modification I just told you about. So we can't assess a variant effects in this protein using like a phage or yeast display assay. And so that's the concept of multi-step. So here's just what a little bit of data looks like. So this is staining of either control cells displaying wild-type factor IX in blue, or our factor IX library containing all 10,000 missense variants of the protein in red. And you're just looking at the, a sort of density plot, a smooth histogram of all the cells in either the control or the library and their level of fluorescence, which is to say their level of factor IX on the surface of the cells. And so you see the control cells have a nice high level of factor IX, and our library has some cells that are like the wild type control, but many other cells that express variants that are lower abundance on the surface and thus poorly secreted. And so what we did was then use fluorescence activated cell sorting to sort our library into 4 different bins that are shown here, a sort of low fluorescence and thus low secretion bin, a medium low, medium high, and high fluorescence and thus high secretion bin. Once we'd sorted all the cells out, we sequenced deeply each bin, and then from the distribution of each variant across the bins derived a functional score, a secretion score, for every variant in the library as read out by this antibody. And so we get a sequence function map that looks like this, where again, every column is a different position in the factor IX protein, every row is a different amino acid substitution. And collectively, this heat map tells you the results of every possible missense, or 98.5% of the possible missense variants of this protein. So what you immediately notice are these big blue stripes. Those are positions that are exquisitely sensitive to any variant. So any variant there prevents secretion of the protein, and we can understand why this happens if we look at the structure of the protein. So these are structured regions, often buried in the folded cores of the protein. And if we make a variant there, then the protein doesn't fold and it gets recognized and degraded by quality control machinery, so it never makes it out of the cell. This heat map has many interesting features and surprises if you want to understand factor IX biochemistry and even think about engineering factor IX to make recombinant factor IX more stable or a better drug. You can understand, for example, in the signal peptide region, how variants either suppress or slightly enhance secretion of the protein, and the same is true in many other functional regions of the protein. So there's lots of interesting things to say about this heat map, but I don't have that much time. So I'm going to skip right to the clinical part and just sort of answer the question for you. What can we learn from this data relative to people who have hemophilia? And so one interesting thing about hemophilia is that the clinical community has done a great job of phenotyping a large number of patients and carriers of factor IX variants. And so we know for several hundred variants what the circulating level of factor IX in the plasma is, and that's shown on the x-axis of this plot. And so we can ask, how does our sort of humble assay in cultured human cells, where we can assess the level of secretion of each variant, correlate with the levels seen in human plasma? And you might, like, one hypothesis would be that the correlation should be pretty good, because the main determinant clinically of the main molecular determinant of the amount of factor IX we see in each patient is going to be the production, the secretion of the variant. And another hypothesis would be, no, that's not going to be the case. Instead, the main determinant of the amount of factor IX in patient plasma is in fact going to be something else, like the sensitivity of the variant to plasma proteases, or its ability to be cleared by some other mechanism. And what we see is a quite striking correlation with our functional data from our in vitro assay, where, if the variant is poorly secreted in our assay, the patients also have almost invariably low factor IX levels, and likewise, if the variant is secreted well in our assay, then most of the time, almost all of the time, patients have high or normal wild-type-like levels of factor IX. So said another way, our assay has a high predictive value for this patient endophenotype, which is useful if you want to think about understanding why variants are pathogenic. Additionally, hemophilia is classified as either mild, moderate, or severe, and this has important implications for how patients are surveilled and treated. And what is shown here is the assay scores from our assay, so just the amount each variant is secreted in each of the three categories for many patients. And what you can see is that severe patients have a really large enrichment of very low or null secretion scores in our assay. Or said another way, if you have a variant in our assay that is very poorly secreted, you have a much higher likelihood to have severe disease. And again, this is really useful if you want to think about understanding what a newly discovered variant might do. Lastly, we use this data to reinterpret variants in the My Life, Our Future hemophilia genotyping cohort. And in that cohort, about half the variants were variants of uncertain significance when we began our work. And we were able to reclassify about 60% of them to a more definitive, likely pathogenic interpretation. And again, these results were returned to patients and who now have a molecular diagnosis. So that's what I wanted to say about developing a method for secreted protein variants. And I want to use the rest of my time to tell you about another technology we've developed for directly measuring enzyme activity at scale. And that's something we also couldn't do before. So, this is going to be no surprise to this crowd, but loss of enzyme function is an important mechanism of pathogenicity of variants. And like I said, we didn't have a way to directly measure that in a multiplexed fashion previously. And so we decided to work on such an assay. And for this work, we picked cytochrome 2C9 or CYP2C9, which is a protein that it's an important pharmacogene because it metabolizes a variety of different clinically highly used drugs. Perhaps the most frequently used one is warfarin, an anticoagulant, and different CYP2C9 variants can either turn over warfarin in an enhanced way or a reduced way, and that can alter how much warfarin a patient needs. And because underdosing or overdosing this anticoagulant can have can cause either thrombosis or bleeding, getting the dose right is important, and there is a desire to use genotype information to guide dosing. So what we wanted to be able to do was convert a sort of well-validated one-at-a-time yeast assay where human CYP2C9 variants are expressed in yeast, and then their function is assessed using a fluorogenic substrate, so a substrate that is, that is turned over by the enzyme. It becomes fluorescent, and then you can measure the fluorescence. We wanted to convert that into a multiplexed format. Seems like a simple idea, except the fluorophore leaks out of the cells, so you can't mix cells that have different genotypes together. But our vision was to be able to somehow make the fluorescence of each yeast cell proportional to the activity level of the enzyme that it expresses, and then play our same trick of sorting a library into bins, sequencing each bin, and scoring every variant in the same way that we did in the multi-step assay I told you about. It all sounds good, but it was hard to do. The way we solved that problem of the fluorophore leaking out of the cell was by using an activity-based or mechanism-based inhibitor of CYP2C9 or substrate of CYP2C9, where when the enzyme reacts with the substrate, a covalent bond is formed, so the substrate, the product can't diffuse away. And then we use click chemistry to add a fluorophore. So now the covalent enzyme product complex is fluorescent. And that took a lot of work to make it work, but it was successful. We used a probe that had been developed in the Wright lab called this. It's called this Taha probe. That's sort of a generic probe for cytochrome activity. And here's what the sort of flow cytometry data look like. So here's the level of fluorescence of yeast cells expressing wild type CYP2C9. Here's the reduced fluorescence of yeast cells expressing a known allele that has about 60% activity, the STAR2 allele. Here's what yeast expressing a further reduced 20% activity allele, STAR3, look like, and finally a null allele, this C432H variant. So you can see that the fluorescence of these controls sort of reads out the level of activity that we would expect from these variants. So we went ahead and made a library, and just like in our factor 9 example, the library sort of spans the range from wild type like to complete loss of function, and we played our same trick of binning this library into 4 different bins, sorting the cells in those bins out, sequencing them, and computing a score. So here's just a summary of what the data look like. So these are just a histogram of the activity scores. Our synonymous variants score up here, 1.0 means wild type in this scale. A nonsense variants, which are complete loss of function variants, have a score of 0. And here's our 3 controls, complete loss of function, 20% activity, 80% activity, or 60% activity, and wild type. And then all the missense variants in the library kind of spread out. Some are wild type like, some are loss of function. So we scored 6,000 missense variants in this assay, and we're able to do a lot of fun stuff with the data. So of course we get another sequence function map, just like for factor 9. There's a bunch of interesting trends in here if you want to know, and patterns in here if you want to know about the biochemistry and structure of the protein. Like we learned a lot about the transmembrane domain and the heme binding motif, which is a critical cofactor for this enzyme, and is very sensitive to mutagenesis. So just to remind you, if you want in the paper, you can read about all the things we learned. But to skip right to the sort of precision medicine related part, we also analyzed all the variants in CYP2C9 that had been identified in the NOMAD database, so just the population sequencing, human population sequencing at the time. And what you can see is, and so what's shown here is just all the variants in NOMAD arranged by position in the protein, and their allele frequency, so high, so these STAR2 and STAR3 variants are the high frequency commonly seen variants, and then all these ones down here are very rare or even singletons. And they're colored according to their activity in our assay. So purple or blue means loss or reduced activity in our assay, and red means wild type like, and yellow means increased in this plot. And what you can see is a kind of rainbow, right? And I guess what I want you to appreciate is that out there in the population are people with profound loss of CYP2C9 activity, some people with increased activity, many fewer, but some people with increased CYP2C9 activity, and some of those are the well-known star alleles that have been studied by the pharmacogenomics community for a long time. But a large number of individuals also carry rare or ultra-rare variants that have never been seen before, have functional effects, and presumably will alter these patients or these individuals' response to drugs like warfarin. And so hopefully this plot gets you to think about how having this data might alter how those patients are treated. So anyway, just to sum up, we analyzed functional effects for about 400 variants in CYP2C9. We also have a map for VCOR, another pharmacogene, where we resolved questions about warfarin resistance and the protein structure, and a map for CYP2C19, where we provided functional data for another 400 human variants. So the vision of the lab is that we should collect functional data for every variant in every disease-related gene in the genome for many different phenotypes. And I've tried to give you a flavor of how we can use molecular phenotype information to understand human disease. Doing this isn't something my lab alone can do. So we have a database, MAVEDB, that has about 8 million variant effect measurements if you want to go find these data and use them. There's also the Atlas of Variant Effects Alliance, which is a place for people who are interested in this idea to connect and plan work and share strategy. We also have a conference, the Mutational Scanning Symposium, that is in its 7th year that you could check out if you're interested. So with that, I just want to say thanks to the people who did the work, in particular Sean Fayer, who led the clinical variant interpretation work that I showed you, Nick Pop, who's not on this slide because he went back to medical school, who devised multi-step and the Factor IX system, Claire Amorosi in Maitreya Dunham's lab, who did the CYP2C9 work along with folks in my lab, and you for your attention, and of course our funders. Thanks.
Thank you very much, Dr. Fowler, for your excellent presentation, and to all our speakers in this session. So with our remaining time, we'll move to Q&A portion of the session, and for those in the room, please step up to the microphone to ask your question, and for our online attendees, please use the Q&A box in Zoom. And prior to asking your question, if you can state your name and affiliation. Thank you. So I have a question in the chat box, and this is for Dr. Stefan Kemp, and this is from Arash Unisi. Genes usually have pathways of dozens or hundreds of genes that affect each other's RNA expression. Are there studies on the pathway containing ABCD1 to compare them between the different types of patients?
Yeah, that's an excellent question. This is, well, we have not yet performed RNA-seq, and I don't know if any other of my international colleagues have done it. And if it's been done, it's certainly not for a large cohort. So, yeah, for the sake of time, the short answer is that it's a good question, but I don't have the answer. But it's likely to happen.
Thank you.
I had a question for Dr. Theodorescu. This is Bob Shook from the Division of Translational and Precision Medicine in the Center for Drugs at FDA. Just curious if your multi-omics approach in creating the digital twins incorporates germline genetic testing for pharmacogenetic variants, such as if a patient was eligible for like bifluorouracil treatment, would they be tested for DPYD, and would that be incorporated into the algorithm?
Yes, thank you for that question. You know, I didn't have enough time to mention all the things we included. We did not include that in the original paper, the one that just came out. We do have that for follow-up. The nice thing about the twin architecture is that it's modular, so you could just keep putting stuff in. So we have that in the next version. The other thing that I didn't mention, that we're doing this now, literally we're writing this up now, is we have computational radiology integrated into the molecular twin. I want to do that in the original paper. They did not, I could not make it happen in time. So we only had computational pathology. Now we have computational radiology. So again, it's going to refine what the clinician has to make the surgery, no surgery decision. So we'll have computational pathology, computational radiology, and all the blood to make that decision. Or sometimes we don't have the biopsy, but so we'll have that as well. So there are pieces like that. And of course, the other sort of big elephant in the room type analyte is microbiome. And we don't have that either integrated, but we've collected all the samples. So a lot of exciting times ahead. But the machine, I've been impressed of how, now that we have the structure set up, how easy it is to integrate an analyte, whether it doesn't matter, the machine doesn't care what the analyte is. And so once the programming is involved, it's all good.
Thanks. It's great to hear that you're continually improving the algorithm. Thank you.
And a question for Dr. Fowler. Thinking about the Alliance, do you envisage a time or a time frame for the atlas to be completed for all codeine genes or just all clinically relevant codeine genes?
Thanks. That's a great question. I mean, I think we're at the point now where we have functional data collectively, the community, for perhaps 100 human genes. So well short of 4,000 or 5,000, which would be the sort of clinically relevant set. And I think, you know, if we continue business as usual, I would say in perhaps 10 years, we'll have most of those 5,000 done. But what's exciting is that there are some large initiatives that are kind of just getting started. The Impact of Genomic Variation on Function Consortium that the NHGRI has funded, as well as others that I think will accelerate that. So I would guess that we'll have many data for many of the genes that clinicians will care about, like let's say a set of perhaps 1,500, that would be like GenCC genes, in something more like 3 to 5 years. There are also some interesting things happening, like for example, we are about to federate our NaveDB database with ClinVar, so clinicians will be able to discover variant effect data along with the information they need to use it in ClinVar rather than having to like know about it and go to our database. ClinGen is also releasing rules that will help better define how to use functional data. So I think that in the 3 to 5 year time span, a lot will happen. I don't think we'll get completely done in that time span, though.
Great, thank you. And we had a question from Vivek Dash, but I believe Dr. Theodorescu, you answered that directly to him. Is that correct?
Yeah, yes, I did.
Great.
I'm happy to say what I answered if it's of interest to the group, you feel.
Yeah, please.
Okay.
Unfortunately, the question disappeared.
No, I could see it because there's a section answered questions. So basically, the question was, can you basically, it's a lot of analytes, 400 plus, and can you reduce it? My answer was, we've optimized it further. We have about 100 and almost 200 now, but given the technology that we use in the mass spec lab, and the ability of that lab to have commercialized multi-protein assays before, we think that 190 is very easily doable. And as I showed you on the cartoon on the slide, it's also amenable to offsite monitoring with the Mitra device or other similar devices. So I think we're going to try to do it with 190, and that's also after optimization for the computational imaging. So that'll be done in context with the computational imaging as well as a package. Yeah, so I think it's doable to do it at that level.
Thank you. Great. Well, that ends our session for today, and I'd just like everyone to put their hands together again to give a round of applause for our speakers in the session today. Thank you. So we're going to break for the poster session and lunchtime. The posters are in the room next door to the left, and lunch items can be purchased at the kiosk outside the room to the right. And I look forward to seeing you all back here at 1:15 p.m. for our afternoon breakout session. Thank you, everyone.
And just a quick reminder.